make that available on the KTN's YouTube channel. And finally, this webinar is perfectly timed that my local ice cream truck will probably interrupt me. So if you hear that, we'll just pause for about 10 seconds while he plays his music and then continue. So I'm going to give you now a brief introduction of KTN and the Microbiome SIG. So KTN is the network of networks in the UK. We have over 160 sector experts like myself supporting our enormous network of contacts, which are mostly industrial and academic within the UK. We're primarily funded by Innovate UK, so a government organization which is part of UKRI, which is the UK's research councils. Here, bear with me 1 second. Always great to have the local ice cream truck. So essentially, Innovate UK funds KTN to support business. So we support primarily, well, almost entirely R&D focused business. We help them access both the research base as well as the technology base to improve the speed of R&D and help to capture more value. And so how we help businesses essentially is we help businesses partner. So we help them access both industry partners as well as academics. We help them with funding. And so this is a major portion of why people know us. So when it comes to Innovate UK funding, we can take a look at your applications. We can help you understand the funding, etc. We organize quite a few events. So traditionally, again, these would be in-person events, but of course now more and more online. And we attend events to help share kind of information about both funding and other opportunities that are out there. We do a lot of one-on-one work, particularly with businesses in the UK to kind of help them enter this ecosystem. And finally, a lot of what we do is kind of this strategic work, which is taking industry views and making them accessible to all stakeholders. And that's where this special interest group fits. So I'm just going to fly through a little bit about KTN in numbers, but you can always come back to this slide. So as I say, about 160 sector experts. We have a huge number of contacts across the UK in almost every sector that operates here. So that could be anything from space to digital and creative, sensors, agri-food, health, biotechnology, etc. We organize a lot of events, and in general, businesses find it very beneficial to work with us. And that's great because we are a free service. So please use us if you need to, and check out the website just by searching for KTN UK. So just a little bit more about the special interest group itself. So these special interest groups are groups that are funded by money, which is ring fence given to us by UKRI to support areas of high potential. So where the UK has the capability or the capacity and potentially could succeed if there was more attention and a larger community in this space. And we felt microbiome fits this very clearly. I think the UK has both the underlying research capability as well as the industrial capacity to deliver in this space, but we're maybe not seeing as much activity as we would expect. And so we pulled together an industrial advisory board comprised of people across different sectors that are operating within the UK to help us focus on what we want to accomplish with this special interest group. And kind of a brief version of what the goals of this group are, I'll run through now. One is, as I kind of mentioned, is to build a proactive and self-sustaining community within the UK. And that's something that we want to have comprised of both face-to-face opportunities to network, understand what other people are doing, but as well as things like this webinar and other virtual events so people can connect, you know, either if something is too far away or, of course, during this COVID-19 pandemic. And so that's a major portion of what we're trying to do. And if you have any ideas or want us to help promote any of your activities that you feel like fit these areas, feel free to reach out to me. Secondly, we want to raise the profile of what's happening in the UK. So the world leading science as well as the industry activity. And again, we feel like these webinars fit that purpose, but we're also working on the landscape map. Many of you will have heard me talk about that before, but that will highlight both industrial and academic activity in the UK. And we hope to have that live fairly soon. And I've said that before, but we have the map done. We just need to get it out there for you to see. Goals 3 and 4 are kind of related, and we definitely want to support the community in creating more spin-outs and startups in the microbiome space. So commercializing a great technology, finding the investment they need to both get moving as well as to scale up. And we think that's very important. And some of the activity that will happen probably next year will focus very strongly on this. And then finally, we're very interested in forming UK government policy where appropriate, to help them understand where the opportunities for investment are, what some of the challenges are, and where maybe infrastructure is needed by the community. And we currently have a process going to create what we're calling a strategic roadmap for the space, where we have 12 work streams across many of the different sectors that are interested in the microbiome, as well as some of the cross-cutting themes, with over 70 key opinion leaders. And we hope to publish this document, which will highlight some of these opportunities and focus areas in the fall, probably end of September, early October. So there's a lot of other activity. Please feel free to check out the website, which is at the bottom, or email myself or our brilliant chair, Andrew Morgan. If you have any comments, want to get involved. So as I say, sign up for our newsletter, check out our LinkedIn, and there'll be more opportunities such as this webinar. And we just want to get involved with you as much as we can. So that is it from me. I'm going to stop sharing my screen now, and I'm going to pass you over to our main speaker, Jethro, who will take it from here. So Jethro, please go ahead. You're muted still, Jethro. There we go.
Just worked that out. Thank you. Okay, thanks very much, Charles. And thanks also for the opportunity to talk today. I'm really pleased to be part of this community. And in terms of today's presentation, I'm hoping that the knowledge transfer will be a two-way street. And so I'm looking forward to the chat afterwards as well, and to all your questions. So to give you a brief overview of what I'd like to talk about, I'd like to take just a second to introduce the Center for Microbiome Studies. And then, because I'm aware that this is likely a broad audience, I'd like to spend a little bit of time talking about the 16S gene, which is obviously fundamental to this presentation, before going on to present some recent results and talk about the 16S gene for species and strain level taxonomic resolution in microbiome studies. And then I'd like to end with some conclusions and future perspectives that will hopefully lead into the chat. So to begin with, my official position at the moment is as Deputy Director of the Oxford Centre for Microbiome Studies. We're a relatively new research facility that's based in the Kennedy Institute of Rheumatology here in Oxford. As a research centre, we're able to collaborate with microbiome researchers across the whole of the UK. And in doing so, we offer support for all aspects of microbiome research, from experimental design through to bioinformatic analysis. And this is supported by microbiology facilities, so aerobic and anaerobic culturing. We also have access to state-of-the-art notibiotic facilities and germ-free facilities. We're able to support both sequencing and metabolomics, and of course we have access to high-performance compute capacity as well. So I'd just say, if you are interested in collaborating, or if really you're just looking for support in terms of microbiome research, then please do get in touch with us. But this talk itself is based on a fairly simple premise, which is simply that the strain is the functional unit of the microbiome. This may seem like a really obvious statement, that closely related strains of bacteria have biologically important differences, both in terms of their genome, but also their function. And thus, to understand relevant differences in the microbiome, you need to be able to distinguish between different strains in your samples. And really, I think the question that I'm trying to address here is if and how this is possible. And of course, I'm doing this while being explicitly focused on 16S ribosomal RNA gene sequencing. And as such, of course, this whole talk is focused on the aspects or those components of the microbiome that actually possess a 16S gene. But I borrowed this analogy from Professor Mark Adams at the Jackson Laboratory for Genomic Medicine. And Mark has always compared looking at the microbiome using 16S RNA gene sequencing as like trying to identify the make and model of cars from looking at their steering wheels alone. I like this analogy, but I'd also extend it by saying that if 16S is like looking at cars based on their steering wheels, then shotgun metagenomics is perhaps like trying to reassemble cars from a junkyard. And I'm sure that many of you have opinions about the relative merits of 16S versus shotgun sequencing, and I anticipate perhaps this will be a topic for discussion. But really, for my part, I'd say that both approaches have their own strengths and weaknesses. But I would reiterate that this talk is explicitly about 16S sequencing. And as I said, so being mindful of the fact that there's a broad audience, I'd like to take a little bit of time to introduce the 16S gene itself. So this is something that's found in all bacteria and some archaea, and of course it includes the small subunit of the ribosomal RNA. And the nature of the RNA structure, of course, means that you have highly conserved regions interspersed by highly variable regions. And this, of course, is reflected in the gene sequence itself. And so what I've done in this plot on the right is taken several thousand 16S genes from a public database, aligned them, and then calculated the variability or entropy at each base position along the gene. And so what you can see here along the x-axis, which, apologies, I haven't labeled, is the position along the roughly 1500 base gene. And the y-axis is the standardized entropy at each base position. And the shaded regions here reflect the approximate locations of the 9 variable regions in the gene, in this case, picked out by primer positions. And of course, the utility here is based on the fact that the conserved regions are really optimal for primer design, whereas the variable regions contain enough sequence variation such that you can distinguish between taxa of interest based on their sequence alone. Now, I'm not a historian, and use of the 16S gene predates my professional memory, but it's worth noting, I think, that approaches to 16S gene sequencing have changed significantly over the last few years. And I've cribbed the first part of this from an early review, but it's simply to say that originally it was possible to amplify the full 16S gene, the amplicon being shown here in red. And if you're using Sanger sequence, which can produce 700 to 900 nucleotides sequences, then you could either sequence a large chunk of the 16S gene, or by doing paired sequencing and overlap, you could cover the full gene. And of course, with the advent of commercial sequencing in around 2003, and really the dawn of high throughput sequencing, one of the major constraints is the size limit of the sequences that you can reliably produce. And therefore, it suddenly becomes necessary to amplify and sequence only subregions. And in this little cartoon here, I've shown the variable region. You could sequence, say, V1 through to V3, or indeed V3 through to V5. And so, of course, in around about 2007, when the Human Microbiome Project and comparable studies were kicking off, if they were using 16S, they were sequencing on 454 and targeting these regions. So arguably today, Illumina is still the dominant platform for 16S sequencing. And while it has paired end sequencing as an option, such that you can merge complementary sequences to cover a larger region, still you're restricted to targeting sub-regions of the 16S gene. In this case, I've depicted V1, V3, or even just a single variable region in terms of V4. And of course, now with the advent of 3rd generation sequencing, it's possible to amplify and then, for the first time in a high throughput manner, sequence the entire 16S gene. So to give a retrospective view on how 16S sequences are handled, first, of course, as I've just said, you amplify your variable region of choice and sequence it on your platform of choice, and if necessary, merge your paired end reads. But then traditionally, it's been typical to cluster sequences, i.e. sequences above a particular threshold are collapsed. So in this 2D representation, you can see down here, each little data point is a single sequence, and the distance between two sequences represents their percent similarity. What you can see is that highly similar sequences would be collapsed into a centroid, so there would be one representative sequence moving forward. And most commonly, sequences are being collapsed if they are 97% or more similar across the length of the amplified region. So practically speaking, this can be done to remove artifactual error introduced by PCR or by sequencing. So for example, one of these little red data points might have sequence errors at one or a few nucleotides. Those would subsequently be removed from the collapsed sequence. And while it may not always be desirable to collapse closely related taxa, it more or less stands to reason that you can't distinguish between two taxa if the sequences of those taxa diverge by less than the error on the platform that you're sequencing on. So this is something of necessity. So the resulting sequencing, the resulting centroid sequences are, of course, termed operational taxonomic units, and that name has a certain level of ambiguity when it comes to referring to the actual taxa that these represent. And of course, I think that that ambiguity is very important. And for me personally, this where the study that I'm going to talk about today started. There may be, as I've said, very valid reasons why you would want to collapse 16S gene sequences, but I've been left wondering in the past why it is that 97% is an adequate threshold, and what this actually means in terms of the two taxonomic resolution that you can actually achieve from 16S gene sequencing. So again, I'm not an historian, but my brief literature search settled on this paper, which is by Stackerbrandt and Ibbers, and I think it came out in 2006. So really what it shows you is how well the 16S gene can act as a proxy for DNA-DNA hybridization across the genome, which for the purpose of this talk, we're going to assume is the gold standard for determining whether two strains in a lab are in fact the same species. So essentially what the paper is saying is that if two bacteria are greater than 70% similar in terms of their DNA-DNA hybridization, you can consider them to be the same species. If they're less, then they're likely different species. And so each data point on this plot is actually a pairwise comparison between two strains, where, as I've said, the x-axis is DNA-DNA hybridization, and the y-axis is the percent similarity of the 16S gene. And so, of course, just to reiterate, what this shows is that greater than 70%, you very seldom see, or in fact never see, a 16S gene that is less than 97%, this red bar here, or indeed in this 2006 paper, 99% similar. Thus, if you sequence the 16S gene alone and you found that it was less than 99% similar, you could be confident that you're not dealing with the same species when comparing two different strains. So here we go. To reiterate, essentially, you never see two species where the 16S gene is less than 97% similar, but they would be considered to be the same species by this metric. But, and I think it's an important but, on the top left here, what you can see is that there are many cases where you see two bacteria where the 16S gene is greater than 97% similar, but in fact, based on this metric, you're dealing with two different species. So to put this back in the context of high throughput sequencing, there are many situations where if you were collapsing at 97%, you'd be collapsing different species into the same OTU. And I think there are two important things to take home here. The first is that for the last few decades, at least, I think many microbiome papers have been guilty of an inversion fallacy, simply saying that if two sequences are greater than 97% similar, then they likely come from the same species. Whereas in fact, if you dig back in the literature, there's really no evidence of this. And the second point here is that taxonomic resolution afforded by 97% is not really getting towards our holy grail of species or strain level resolution. And so really for the rest of this talk, I'd like to go on and address the idea that what we're trying to do is break this 97% barrier. And really ask what taxonomic resolution can the 16S gene actually give. And so for the rest of this talk, with that in mind, I'd like to really address three different points. And the first is how well can different variable regions actually resolve species? So if we're limited to sequencing only part of the 16S gene, can we get to species, or, and then obviously from then on strain? The second point is really to look at PacBio circular consensus sequencing as an approach for full-length 16S gene sequencing. And finally, I want to give you what I think is compelling evidence that you can use intra-genomic 16S gene copy variants as a potential mechanism to resolve strains of the same species. So to start with, I would ask, how well do different variable regions resolve unique 16S gene sequences? How well can you use different variable regions to get to this species level comparison? And this is really reporting an in silico study here. This is the same entropy plot that I showed earlier, but on the bottom, you can see combinations of variable regions that are commonly amplified using available primers. And these are the variable regions that I'm going to go on to discuss. And they, of course, range from the potential amplification of the full V1 to V9 region, which I'll refer to as full-length sequencing, to the amplification of the shortest region here being V4. And so as I've just alluded to in my introduction, a sequence-based definition of a species is a complicated thing. So to simplify this issue in my in silico comparison of different variable regions, I set an arbitrary definition to benchmark against the full 16S genes. So what we really did in this case is I went to a public database and took 14,000 available sequences or thereabouts. and clustered those at 99% similarity. So really what you have in the end is a set of sequences that are non-redundant, and each sequence differs by at least 1%. And I use this as a working definition of what constitutes a species. And then in silico, using available primer combinations, you can essentially take this original database of around 14,000 sequences and trim it to make comparable databases containing just the variable regions of interest. And then it's just a question of asking the simple question of what, on this basis, how much information is retained in each of those variable regions, and thus how many of those variable regions can still identify the original sequence from which they were generated. And I did this by comparing the sequences back to the original database using a standard classification approach. And the answer is interesting. It's essentially, what you can see here is that different variable regions vary greatly in their ability to discriminate what we know were 16S sequences that differed by at least 1% across the full length of the gene. And so in this plot, what I essentially did was build a tree based on the taxonomy of each sequence in the in silico database. And I'm showing the same tree for each variable region, but the take-home message is that the darker the branch, the greater the proportion of sequences that fell within that particular branch that could not be matched to their true original sequence. And so there's two things that pop out here. One is that the V4 region alone performs particularly badly, but also I think that's a more subtle point and perhaps isn't really visible on this plot, but certain branches, certain variable regions perform better and worse in certain branches of this tree. And so the take-home message there would be, if you're interested in particular taxa and you have to sequence variable regions, be careful about which variable region you choose to resolve those taxa. So perhaps to give a more succinct summary of this result, I'm showing just a bar chart here, which is just a simple plot of the percentage of sequences that could be confidently identified as matching the sequence of origin, which I was using as a proxy for species. And again, you can see that this is set against V1, V9, i.e. the full 16S gene. But again, V4 performs particularly badly in this test. And another more simple way to reiterate this point is to just take those in silico generated variable regions and recluster them using an OTU clustering approach and see how many OTUs are generated by each variable region. And in this case, the dashed line is the actual number of unique sequences that were in the original database, and each bar shows the number of OTUs that can be regenerated using a 99% identity using each variable region. And so to conclude from this, I'd say that there are real and significant advantages to sequencing the full 16S gene over sequencing just variable regions. But I think the other point to make here was that actually in this in silico study, at least variable regions covering V1, V3 was the next best region to consider, at least in this example. And I think that practitioners will know that there are practical problems associated with sequencing V1, V3, and that it's quite a large region. And even with 2x300 paired end reads on Illumina MiSeq platform, it's quite difficult to overlap confidently your paired reads to cover this full region. Nonetheless, we took V1, V3 and V1, V9 forward and tried to ask whether the results we're seeing in silico are true. Are there practical advantages to full-length sequencing of biological samples, actual biological samples? And so, as I said, we took the best performing variable regions, so full length being V1, V9 and V1, V3, and compared them on some human microbiome samples. And of course, because Illumina can't cover the full 16S gene, in this case, we did the V1, V9 sequencing using PacBio. And I should note, this is on the RS2 platform. So in this case, we used samples that came from a colleague of mine who was actually looking at professional athletes. So these human gut microbiome samples taken from downhill mountain bike races. And each sample is named after the bike of the donor. So really, to simplify the comparison that I'm showing here, we're reporting results only for a single genus, and this is Bacteroides, which is a useful genus because it's ubiquitous in the human gut microbiome, but it's also quite diverse at species level. So it's an ideal genus to choose for a targeted comparison. And what you can see on this plot is that the estimated relative abundance of this genus in the 4 samples based on V1, V9 sequencing on the x-axis and V1, V3 on the y-axis. And so you can see they're broadly consistent in terms of the estimated relative abundance of this single genus. So at genus level, both approaches perform well. And for those of you who are fans of enterotypes, it really falls out that 2 of these individuals could be considered to be in one enterotype and 2 in another based on relative abundance of Bacteroides. The difference begins to appear when you start looking at species level. And to do this, I chose to cluster both the V1, V3 and V1, V9 sequences into OTUs at 99% similarity. And actually this performed remarkably well at resolving different species of Bacteroides, but as I alluded to in the Staggerbrandt and Ibbers paper, it isn't guaranteed that this clustering would allow you to resolve species. But in this example, we took our 99% OTUs and we aligned them to a reference database of all known Bacteroides genomes and used metagenomic whole genome shotgun sequencing of the same samples as a benchmark or the gold standard for assessing the true relative abundance of each of the species in these 4 guts. And so this plot only shows the 3 most abundant Bacteroides species for each of the 4 athletes in this study. But what you can see immediately is that when you're looking at species level, the diversity is much greater than anything that you perhaps would have anticipated by just looking at genus, but also that there are inter-individual differences in the dominant Bacteroides taxon within each gut that also are perhaps biologically relevant. And so this really highlights the advantage of being able to look at species level. The second thing to note is that actually, if you're looking at the predicted relative abundance from these 3 different sequencing approaches, V1, V3 did remarkably well at this clustering level, but it's worth pointing out that there was one species, in this case Bacteroides intestinalis, that you just could not detect. And this is shown in Breezer here, the first sample that had relatively high abundance of Bacteroides intestinalis on the basis of shotgun and V1, V9, but V1, V3 clearly misses this species. So again, I'd argue that this gives you evidence that there is a significant advantage for full-length 16S species sequencing, sorry, for species level resolution. And so having demonstrated that there's a theoretical and to some degree practical advantage for full-length sequencing, I think it's necessary to benchmark the sequencing approach used. And certainly there are many others who have done this with PacBio sequencing. But of course, as I said, it's necessary because you can't seek to resolve taxa if those taxa have sequences that diverge to a degree that's less than the error rates on the platform that you're using. So again, because I'm talking to a broad audience, there may be those who are unfamiliar with PacBio circular consensus sequencing, but the idea here essentially is that having PCR amplified your 16S gene, you would put adapters that would separate the Watson and Crick strands and circularize them such that when sequencing, you can just sequence around the circle in a continuous manner. So you actually cover both strands multiple times. And then what you're left with is an alignment of the strands such that you can generate a consensus sequence, or the circular consensus sequence in this is the terminology used. And assuming that the sequencing errors across this region are random, then the consensus should actually be able to give you a highly accurate sequence. And so we did this by sequencing a mock community full of, which contained 36 species and 24 genera. And the first thing we did, of course, was look at the number of passes, so the number of CCS passes, the number of times you have to go around this circle in order to minimize the error, the observed error in the sequences generated for this mock community. And what you can see here by looking at the total error up in the top left is that 10 or more passes, and again, I should point out that this is on the RS2, so this is an outdated technology now because PacBio have replaced this with the SQL and SQL2. But you can see that 10 passes was sufficient to minimize total error in these reads. And it's also noteworthy that the residual error here wasn't due to deletions or insertions, but it was actually substitution error, and this is something that I'll come back to. Thus, we conclude that PacBio looks extremely promising as a way to generate highly accurate long reads for 16S gene sequencing. But there was one important caveat that I think is reminiscent of issues with 454, in that we noticed there was an issue with homopolymer runs. So if you get runs of the same base in your sequences, that coincided with deletion errors in the reads. I mention this now because from this point going forward to circumvent this issue, I just didn't deal with insertions or deletions in the analysis. This isn't an ideal solution, but I don't think it invalidates subsequent results. So I think this is where this talk becomes slightly more interesting. Having established that CCS is a viable approach, we went one step further And I alluded to the fact there are substitution errors that appeared in those reads. And this was, in fact, it turned out because we were aligning each of the sequences in our mock community. So we had 36 species, and we were aligning each of the sequences to a single representative 16S gene per sequence. And of course, this belies the fact that there are multiple 16S gene copies in some, but not all, bacterial genomes. And therefore, what we were actually picking out as being alignment error may be legitimate variation between 16S gene copies that exist within the same genome. And so this is just to reiterate that point. On the plot on the right, you can see the position and relative frequency of alignment errors that were occurring in all the reads that were aligned successfully to E. coli, being one of the single species in this mock community. And in this case, this is K12 MG1655. And strikingly, of course, the majority of these alignment errors are non-random and they fall within variable regions, which is quite interesting. So actually there are 7 16S genes in MG1655, and the dashed lines on this plot indicate the relative frequencies of those 16S genes that you might expect if these so-called alignment errors were in fact nucleotide variations. So at one or few of the, that existed in one or few of the intragenomic 16S gene copies. And of course, we know the true sequence for each of the 16S genes within this particular genome. And thus we can predict the error frequencies that you'd see in your alignment if these alignment errors were in fact reflecting true intragenomic variation in the 16S gene. And what you can see here is just a little cartoon of just that. You can predict exactly where you would expect to see errors, and you can predict the frequency. And so this is the predicted plot on the bottom, and this is the plot we observed with PacBio CCS on the top. And so really, I, on this basis, conclude that with adequate handling of long read sequences, you can actually get sufficient accuracy to resolve intragenomic 16S gene copy variants. And I think this is exciting, at least in principle, because if you look across different strains of E. coli, you can see that they may vary in terms of one, but not necessarily all of the 16S gene copies in the genome. And in this example, I create the theoretical error profile for 2 different strains of E. coli. So the original MG1655 plot on the top here, and this is 0157 Sakai on the bottom here. And you can see that if you look at the profile, you can actually see differences between these strains on the basis of the 16S gene alone. So to put it simply, if you expand your comparison across all the 16S genes in a particular genome, you actually increase your chance of detecting subtle nucleotide variations, what would have once been called oligotypes, that have the potential to resolve closely related taxa. And so having observed this in a controlled mock community, we really wanted to see whether you can detect similar intragenomic 16S gene copy variants in vivo. And so for this example, I'll return to the downhill cyclists here, and again, focus on Bacteroides. And in this case, what we did is we went back to these samples and looked at 2 individuals here, so Scott and Ironhorse, who had what seemed to be a high relative abundance of Bacteroides vulgaris. And as mentioned, we'd clustered these Bacteroides sequences into OTUs at 99% identity and established that this was actually, in the case of Bacteroides, a relatively good approximation for discriminating between species using the full V1, V9 sequences. And this therefore really allowed us to ask the question of whether within the OTU that was generated for Bacteroides vulgaris, there was evidence of variation in the sequences that were collapsed into that OTU that could be indicative of variation between 16S gene copies that existed within this genome. And the answer there is yes, this is exactly what we see. So these are the similar variant profiles generated by aligning all sequences belonging to that single Bacteroides vulgaris OTU for those 2 athletes, Scott and Ironhorse. So this is Scott on the top, this is Ironhorse. And I would argue that these 2 profiles are similar enough to be compelling in terms of us picking out a true signal here, but different enough to be interesting. So for example, it's a little hard to see, but if you look in the second large peak that you see in the variable in V3 region here, it looks almost as though, and I may be stretching the data a little far here, that 2 out of the 7 16S genes that exist in this genome have a variant in Scott, but actually 4 would have the same variant in Ironhorse. And similarly, you can see compelling differences in other variable regions such as V5, and I think it's V7 here. So once again, it's possible to go back to the known full genome sequence for strains relating to this species and see how well the predicted profiles that we would, with the profiles that we would predict if we looked at the 16S genes existing with these genomes, how well those are recapitulated by the PacBio CCS data we were generating from these in vivo samples. And that's what the two plots on the bottom here show. So these were the two of full high quality Bacteroides genomes for Bacteroides vulgatus that were available in RefSeq. And what I've done again is generate the predicted profile, both for this ATCC strain, which is 8482, and MPK here, which is originally a mouse strain. And I think in particular for the case for the ATCC 8482, you can see that the expected nucleotide variation that we see within our samples collected from the human guts actually looks remarkably similar to the profile that we would predict if we were looking at intragenomic 16S gene copy variants in this case. And so there's one last experiment that I would like to discuss in the context of this hypothesis that you can use 16S gene copy variants to distinguish strains. And here I took advantage of the fact that my colleague had gone to great efforts to culture samples from these and other athletes in her cohort. So what we were really thus able to do is return to these samples and take the cultured isolates and barcode each one and sequence each one individually using PacBio CCS, and then look at the variation you get between the sequences generated for each individual strain. And after some fairly stringent quality filtering, in order to remove what I assume is sequencing error, what we were then able to do is search for true single nucleotide variants within these isolates that could potentially demonstrate that intragenomic copy variants could be used to distinguish strains. And in total, we had 381 isolates that were sequenced. When we blast the most abundant unique sequence for each of these isolates, this gave us 58 different species. And rather gratifyingly, again, if you cluster these sequences at 99% sequence identity, you get roughly the same number of OTUs, which suggests that as a proxy for species level resolution across the full V1, V9 region, 99% isn't too bad. Obviously, though, it's not perfect. However, the interesting thing is that because there were far fewer species and OTUs than there were actual isolates, this reflects the fact that there were many different isolates that were collapsed into the same OTU. And 54 out of 61 OTUs, and that's about 90%, had evidence that I would say indicated that they had more than one 16S gene copy variant. And so to exemplify this, I'm showing the copy variant profiles that were generated from 4 of the 381 isolates. So the two on the left were at least identified by BLAST as being Shigella flexneri. And what you can see here is a profile that looks somewhat similar. So in I think it's V7 here, you can see that 1 out of the 7 16S genes in this genome looks like it has nucleotide variations in this variable region that were comparable to those we saw for E. coli, which perhaps isn't surprising. Similarly, when you're looking at isolates that were identified as bifidobacterium longum in this case, you can see there were very few single nucleotide variations, but they were compelling enough to look like they were genuine, but also that perhaps if you're looking at single nucleotide variations across genes, multiple 16S genes, you can identify differences that would suggest that perhaps they come from different strains. And I'd like to end on this hypothesis that you can use these polymorphisms as potentially a valuable way to distinguish between strains. This is obviously ongoing work, and it's something that I'm continuing to look at. But I'd just like to conclude by saying that really variable regions differ in their ability to resolve taxa below genus level. And this has implications both for the way in which we use and interpret different variable regions in the 16S gene, but also in the way that 16S databases are curated. I think the next major point is that intragenomic 16S gene copy variants should and must necessarily be accounted for when looking and claiming to look at species and strain level resolution in high throughput 16S gene-based microbiome analysis. I've consciously avoided talking about exact sequence variants in this talk, but I imagine it's something that will come up in the discussion. I think this is a particular issue for that type of approach. And finally, I'd like to introduce the idea that accounting for intragenomic 16S gene copy variants can provide strain level resolution if the sequences are adequately handled in 16S gene-based studies. And this is analogous to oligotyping, which is something I mentioned earlier. But essentially what I'm suggesting is that you would be oligotyping across all the different 16S gene copies within a genome. And so with that, I'd like to acknowledge the support of my previous mentor, George Weinstock at the Jackson Lab, and also Dan Spackeritz and Lauren Peterson, who helped with this work. And I'd be happy to take any questions if anybody has any.
Great, thank you so much, Jethro, for the talk. And I'm sure everybody is clapping to their cameras and muted microphones right now for you. So everybody, you now have the opportunity to ask questions via chat, which I will do my best to read out. Just correct me if you feel like I have misinterpreted what you're asking. And we already have 2 questions from Christopher Stewart to start us off, Jethro. So the first is asking about 2 questions relating to 16S copy number. So this varies significantly between bacteria. Will copy number potentially skew the data towards taxa with more 16S copies? And if there are SNPs in these 16S copies within a bacteria, would they be classified as 2 different strains?
I think it depends. So I think in this talk, I've raised more questions than perhaps solutions, which I apologize for. But yes, undoubtedly, I think that if you have multiple 16S gene copies and they're highly variable, obviously there are curated databases that attempt to deal with this problem. But the issue becomes when you have bacterial taxa in your samples that aren't available in databases, then it becomes very difficult to resolve differences in copy number. And undoubtedly, that's a bias. I think that the issue of single nucleotide variations being classified as different taxa really depends upon the resolution at which you want to try and separate out sequences in your samples. And so if you are, say, going down a denoising approach and using exact sequence variants, then yes, you would definitely run the risk of not only artifactually increasing the richness of your sample estimations, but also splitting out different sequences that would actually perhaps have originated from the same genome.
Perfect. Any more questions? And Chris says thanks. Give just another minute. Any other questions for Jethro? Here we go. Are there any plans to compare this to shotgun metagenomics?
Yes. As I said earlier, I think that I advocate for both approaches. I think that they have complementary strengths and weaknesses. I have done this in the past. I think one of the issues with shotgun metagenomics, unless it's very carefully handled, is you get a very high false positive rate because it's very difficult to assign short reads from regions that are perhaps homologous between different genomes. So I think with this, where I would take this work is ultimately, I think what you would need to do is improve 16S databases such that you're able to account for differences between 16S gene copies and leverage that information to make subtle judgments about differences between strains. But also you need to validate the fact that the 16S gene copies that we're seeing as being variable in this high throughput sequencing approach are actually related to differences at the strain level in terms of the genome and also the function of the bacteria that you're working with. So I'd say that's where my interest lies, perhaps more than doing a straight up comparison of shotgun versus 16S approaches, but I think it's nonetheless interesting.
Here is a question from Karen Woodward. Have you looked at a comparison with other long read amplicons, such as 16S, ITS, 23S?
No, I haven't, but I know that other people are. And I think there's a really interesting company in Connecticut called Shoreline Biome who trying to leverage ITS regions. And you know, I think it's a really interesting point that, of course, there's no need to stop at the 16S gene if you've got this PacBio or other long read sequencing approaches, and you can generate high quality sequences, then, you know, why stop at 1500 bases? Again, I think the constraint there is that our databases that we typically use for assigning taxonomy are restricted. sorry, I hope that answers the question.
Here's a question from Jose. So even if the gold standard Illumina 16S is not great, is PacBio competitive for standard microbiome studies in which the main outcome is to tell how similar two samples are?
So I don't know if anybody from PacBio is listening, but at the moment, I think it depends on how deep your pockets are. I would say that if you're just interested in community level differences, I don't want to admonish V4 or short read sequencing approaches. I think that they have their place. And so if you are looking at a community level for changes, say, for example, dysbiosis in a gut microbiome in relation to disease, then provided that the resolution that you can achieve with those short read sequences is sufficient for you, then it's certainly adequate. And it's evident from all the really interesting microbiome research that's been published over the previous years that this is an extremely valuable and powerful approach. I think that as we get more into understanding exactly what the role of individual strains are in terms of microbiome function, that's when these approaches will have to give way to longer read sequencing approaches.
Thank you, Jethro. That's great. Actually, a number of questions have poured in, but I think we're going to cut it off there. So potentially you could get in touch with Jethro or myself, and we could try to answer those. But I would like to thank Jethro again for giving the great presentation today, and I'd like to thank everybody listening for coming along. As I say, the KTN Microbiome SIG would really like to hear from you and engage with you if you feel like there's anything that we could deliver that would be helpful for the community. If there's any speakers you want to hear, or if one of you listening would like to present on one of these webinars and be my victim. So let's all give Jethro a round of applause, which you won't be able to hear again. Thank you all, and please take care. So
thank you very much.
That's the end there. Thank you. Bye now.